Alright, so this is the workbook video for B2.1, membranes and transport. It has a yellow front page. And so for your first part, we're talking about like basics of cell membrane, which hopefully is not news. And so what are your cell membranes made up from? They are made up from your phospholipids, proteins, carbohydrates, and cholesterol in animal cells only. And so membranes are also found inside of eukaryotic cells, and so when they compartmentalize your organelles, give examples of organelles where they're surrounded by a membrane. Again, hopefully, because we just talked about cells, this is not like news to you, but your cells that have membranes, your nucleus, it has a nuclear membrane, your chloroplast, mitochondria, endoplasmic reticulum kind of just is membrane. Those are like your key ones. And now we get into the structure of the structure. Your phospholipids. And so phospholipids have hydrophobic hydrocarbon tails. It's time. We'll do red for hydrophobic, blue for hydrophilic. Get out of here. Red, blue. And so your phospholipids are hydrophobic hydrocarbon tails, your fatty acid tails that form the core of your membrane, and then your hydrophilic, your hydrophilic phosphate heads that film like the outside because they're in contact with the water. Again, it's like your ice cream sandwich. Your hydrophilic part, and your hydrophobic. And so how do the hydrophilic heads and the hydrophobic tails orient themselves in your membrane? Let me set this down. Hydrophilic heads face outward towards outside / inside of cell, and your hydrophobic tails face inwards towards each other. And so any time is a great time to label a phospholipid. There's your hydrophilic head, hydrophobic tails, don't forget about our bestie who absolutely loves water. And then has the rain boots on. Ooh. And it's thick because they're fatty acids. And so hydrophilic versus hydrophobic, again, this should be familiar to you because we spent quite a while talking about properties of water and talking about lipids. So I'm not going to spend a ton of time going over this. Essentially, hydrophilic, it loves water. Hydrophobic, it repels water. I think the other key thing to keep in mind that is kind of interchanged in this a lot, hydrophobic things that repel water are nonpolar. Hydrophilic things that love water are polar. All right. Stop. Start. Flip. There we go. And so phospholipids are said to be amphipathic. And so when something is amphipathic, it essentially just means that it's both. And so amphi means both. Like if you think of like an amphibian, it's something that lives like both on land and in the water. Something that's amphipathic, something that's both hydrophilic and hydrophobic. So again, we have our hydrophobic region on the inside because we have our hydrophobic tails. Woo. We have our hydrophilic region on the outside. We have our hydrophilic heads. And so two phospholipid layers arrange to position the hydrophilic heads towards the water, while the tails form a core with a low permeability to large molecules and hydrophilic particles. The arrangement of molecules forms an effective barrier between the aqueous solutions. Because there's water on both sides, having this hydrophobic region is what helps it to be able to be selectively permeable and to keep them separate. But then having the hydrophilic heads is what helps them to be able to be in the water without it like freaking out. And so our next step is to talk about membrane proteins. We're going to spend quite a while talking about membrane proteins. What color is proteins? I guess proteins can be green. And so membrane proteins, your cell membrane also has membrane proteins, which can be grouped into two main categories depending on their location. Which ones? Again, hopefully at this point, this is not like crazy new news to you. We'll do orange and, I guess pink. But you have your integral proteins. And then you have your peripheral proteins. Oh, why did I pick orange? The orange highlighter is dying. It's okay. It'll do its best. So integral proteins, we have it here and here, and then peripheral proteins here, here. And so scooch, scooch, scooch. Our peripheral proteins here, peripheral. Why is this one colored weird like this? And then integral proteins. We have an integral non-transmembrane protein and an integral transmembrane protein. Oh. Okay. Well, I don't really think that's showing up too amazing, but that's fine. And I have a whole mess of things. So let's look at our integral proteins first. So I think the key thing is that, like, the way that I like to remember it is integral has in, they're in membrane, they're like embedded. Your portions of your integral protein are both hydrophobic and hydrophilic, depending on where they are. So, portion in membrane is, oh, I put the stupid color, oh my gosh, darn it. Hydrophobic. So like this part here would be hydrophobic, and like this part here would be hydrophobic. And then portion exposed, or like outside of the membrane, is hydrophilic. So like this bubbly part. I picked colors that are like not showing up. Okay, this is supposed to be like outlined in blue. Believe me, trust me. And then key things that makes these different. So when it's transmembrane, it's embedded in membrane and goes all the way through. When it's not transmembrane, it's embedded in membrane, like doesn't cut through, doesn't cut through. See how it like doesn't go from side to side, it just kind of is like blobby in there. And then your peripheral proteins, I think the easiest way to remember this is that they're like on the periphery. Which is like the edge. Or you could think of like peripheral vision. So like when you look at like the side of your eye, like your peripheral vision. I wrote peripheral with an F. Peripheral vision. It's like on the side. And so they can be on the side of integral proteins, or they can be on the side of the membrane. Peripheral proteins, oh, let me spell it with a PH. Peripheral proteins are hydrophilic on the outside. This allows them to bind to your phospholipid heads and will also allow them to be exposed to the cytoplasm, or like the extra cellular fluid. Again, like when we learned about proteins, we learned that sometimes they have different charges or different polarities inside and outside, so they're at least hydrophilic on the outside. The inside, who's to say? Protein to protein. All right. Scooching right along. So now I have a bunch of different types of proteins. Some of these we've talked about in class. Some of these we haven't quite covered super in depth, mainly just because they're like not as common. I think for this page we'll just do like a rainbow. Why not? Ooh, let me get a backwards rainbow. So we have channel or carrier proteins. And so these are your proteins that span the entire membrane and transport substances. Open, please. So they're really used in facilitated diffusion. They're integral transmembrane proteins. An example would be like aquaporins, like for water. Or like your sodium plus or K plus channels. You know, either one. Close. Next one is protein pumps. And so these are used for active transport. They need ATP. These are your salty bananas. Key example, Na plus, NK plus, pump in neurons. They're also in your kidneys and in your intestines. You also have enzymatic or like enzyme proteins. I guess just enzymes. That's kind of redundant to say enzyme proteins in your membrane. They do a ton of different stuff. There's like a lot of them. I think that the examples that I could think of off the top of my head is that you have let me spell this right. Oxidoreductase, which does oxidation reduction reactions, and then hydrolase, which is used in hydrolysis. Which, if you don't remember hydrolysis, it's time to hit the books. Oh, I can't write it in yellow. Oh, I have 3 left. We're gonna write it in yellow. I'm committing. So, hormone binding membrane proteins. So these are usually used for like cell signaling or like receptors. There's like a ton. So like in general, your GCPRs, they bind like epinephrine. Glucagon, but etc. Dopamine receptors. Tyrosine kinases, which deal with like insulin. There's like a bunch of them. Pick your favorite, write it down. Your glycoproteins, I don't like that this one just says cell-to-cell communication. It really should just say glycoproteins, but whatever. Glycoproteins. So glycoproteins. Woo! They're mainly used in the immune system for like viral recognition, cell-to-cell recognition, things like that. And then lastly, what's the last? Oh, red, duh. So what's the last color of the rainbow? Idiot. Get it together, Jessica. So your cell adhesion proteins. So these are things like your CAMs, your cell adhesion molecules. They anchor one cell to another. You have cadherins, integrins, and like selectins. It's not super important that you know like a ton of differences between those. Just know that they do like slightly different types of adherents. We'll look at like the pictures of them, but like without the names later. So like as long as you know that there's different ones and that they have slightly different functions, like that's really all you need to know. And that there are membrane proteins that can help with cell-to-cell adhesion. There's really like not that much to cover there. So those are your different proteins. These are all the functions of them. These are all the things they do. Your examples. Another kind of cool thing, what would you say proteins was? Green? Is that protein content is variable depending on the function of your cell. So like the more active of a membrane you have, the higher protein content you'll have, which like makes sense. Most of them have like 50%, although sometimes, especially with like organelles, like chloroplasts and mitochondria, they'll have closer to like 75%, which I just think is a pretty neat little funsies factsies. It's on the page. Oh jeez, this is another word that I feel like I'm always saying wrong. Okay, there are pros and cons to have spiral boundless. I like that it lays flat, but it's fighting with me. Please cooperate. There we go. It's a work in progress. All right, so now we're looking at your structure and function of your glycoproteins and your glycolipids. Let's see, what color speaks to me when I think about glyco anything? Brown. Alrighty. Is this the brown? Is this the brown one? I hope that's the brown one. I think. We'll assume. So your structure and function of your glycoproteins and glycolipids. They're essentially like your carbohydrates within your cell. And so they're important components of your cell membrane. They're mainly involved in cell-to-cell recognition and the immune system. Your glycolics, which is like this little frilly maneuver you're seeing here. Oof. This is your glycocalyx. It is only in animal cells. It's just like pretty much like the sum of all of your carbohydrates from all of your glycoproteins and glycolipids, forming like a carbohydrate layer on the outer surface of animal cells. And so it's your carbohydrate rich layer on the outer surface of animal cells. And its like job in life is that it can help fuse adjacent cells, helping stabilize. Oh gosh, how do I spell it? Stabilize or allies? Stabilize? I feel like that's wrong. Tissues. And it's just like all of the, it's like all carbs from glycoproteins and glycolipids. Remember that like glyco, anything just means like carbs. All right, what color do we want? Glycoprotein and glycolipid. I guess we'll make glycoprotein green because it's protein. And we'll make glycolipid pink. We'll make glycolipid pink. I'm in a pink mood today. All right. Where's the stupid pink one? There we go. So glycolipid. Boop. Boop. And then dun, 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 dun. Careful. Then we'll also highlight them in our little picture. Glycolipid in the picture kind of just looks like a carbohydrate. So it's not like you can really see the other part, but it's fine. And then glycoprotein. Oh, it's like Christmas. Glycoprotein. I'm trying this as fast as possible because I know the drawing of the cell membrane is going to take forever. Maybe I just won't do that part. Sure, glycoproteins. Okay. And so your structure for your glycoproteins, they are conjugated proteins. Where the carbohydrates is the non-polypeptide portion. They're basically just a protein, like an integral protein or a peripheral protein with a carb chain on it. They're usually integral, but they can be either. Their function, they do a lot for immune response. Oh, let me spell immune right. Immune response. They do cell-to-cell communication. They help us to be able to distinguish self from not self, which helps pathogens and foreign tissues be identified and destroyed. And then your glycolipids, they're like a short, oh geez Louise, a short carbohydrate chain on a phospholipid. They're like embedded into the membrane. Embedded in and then like stick out. They're in all eukaryotic cells. Eukaryotic cells. They also help with cell-to-cell recognition, cell ID. And then with the, glycocalyx, they also help with like cell binding. Slightly less exciting. Alrighty. I feel like me drawing this is going to take forever. I fully believe that we have done enough practice where you should be able to draw this on your own. I'm going to trust you to draw this on your own, because if I draw this with you, we're going to be here all day. All right. Yeah, because here's the draft version of it, and there's more stuff I wanted to add, but was like, I'll just add it in post. So yeah, that's going to take forever. So I trust you. If you really need help, let me know. but I feel like you can probably handle it. Alrighty, so now we're going to look at active and passive transport. Again, this is stuff that we've been over several times in class, so I hope it's not going to be that big of an issue. Your passive transport, there's no ATP. Your active transport is ATP. I think that the way that we're going to go about this is we'll make all the passive transports cool colors, because ATP in my brain is like yellow or orange. So osmosis, facilitated diffusion, oh, it's green because it has proteins involved. Haha, cute. I didn't even do that on purpose. I'm having too much fun. And then simple diffusion. I guess we'll make passive transport as a whole, what, gray? Where's the weird gray? There's the weird gray. Passive transport, no ATP required. Essentially just various forms of diffusion. Passive transport in a nutshell. Active transport, protein pumps to me, because they're complicated, are screaming red. That's pink. Okay, whatever, Jessica. And then we'll make vesicular transport. I guess vesicular transport will be red. Protein pumps, you escaped this time. And we'll make active transport as a whole, we'll make it orange. No, we'll make it yellow, idiot. Duh. ATP is yellow. I mean, it's not actually, but like, you know, energy, yellow, lightning bolt. Don't question it. Alrighty. So the first one we'll look at in detail is simple diffusion. So simple diffusion across a membrane. And so diffusion is essentially just particles moving. Come on. Particles moving from an area of high concentration to, oh, let's use the dark blue. That's smarter. Low concentration. So like moving with, along, down, any of these words, they all kind of mean the same thing. The concentration gradient. Until equilibrium. Or equal. Those words kind of sound the same. Is reached. And so if we look at our little pictures, we can see that we start with an area of high concentration at the top, low concentration at the bottom, high concentration at the top, low at the bottom, and then it'll move until it reaches equal. So high, low, low. This is equilibrium. And so diffusion, we'll bring it this way. And then diffusion will stop. And they'll just kind of stay. Well, they won't like exactly stay, but like some will float back and forth because particles are constantly moving. But for the most part, like there's not going to be any big movements of them. And so like same here, your particles would move from where there's more across to where there's less. From your area of high concentration to your area of low concentration. So we have low concentration down here. Low concentration here. Oof, this is gonna get kind of ugly. Low concentration. And then we got kind of like middle. Oh, thank God I picked red and blue. This would have been like duty in the middle. Kind of like middle here. And we have equilibrium. And then we have, where's the red? Let me get the real red. And then when it's high, I think like low means go. When it's low, that's where the particle is going to go. Will this please cooperate? Don't tell me the red one's dying too. I'd be very upset. I really like the red one. So, low means go. When it's low, that's where they want to go. Alrighty. Boop. And then here's the weird thing to me. I don't know why this packet does this. It goes from diffusion to then randomly talking about permeability. I have no idea why it does that. Questions I don't have answers for. I guess, annoying, but I guess we'll make permeability orange. So selectivity in membrane permeability. So hopefully you all know that your membrane is semi-permeable. And so simple diffusion through membranes only happens for some molecules that the membrane is permeable for. So again, being permeable means that something can permeate or pass through. And so what type of particles can diffuse are a yes, and then cannot diffuse are a no. How about this, please? And so things that can diffuse are like a yes. Anything small, nonpolar, uncharged, which molecules cannot diffuse? Things that are large, things that are polar, things that are charged. Those bad boys ain't getting through. And so then there's also like rates of diffusion based on like size, polarity, and charge of these different things, and if they're hydrophobic or hydrophilic. And so how they would move based on size, polarity, charge, properties. Okay, so how am I going to break this up? I guess fast would be green, right? Because it gets like a green light. Oh, and then, yeah. Oh, it's going to be like a traffic light. I'm so excited. Okay, so things that go fast. Sonic the Hedgehog. Sorry. So oxygen gas. Carbon dioxide. Ammonia. Water, which I know is a controversial one. And the Y, they're small, nonpolar, and/or nonpolar, and then and no charge. So they're small. Some of them are nonpolar. I mean, water is obviously polar, but it's small enough where it doesn't really matter. Anything small and nonpolar, absolutely. Anything nonpolar and maybe like a little bit on the larger side, absolutely. And then no charge for surezies, that's going straight through. So then we'll look at our slow, our yellow light items. Boop, 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 boop. There's actually a lot of these. Why did I make this section so small? Oh well, it's too late now. So fructose, glucose to some extent, like asterisk, urea, glycerol. Okay, there was not that many. I take it back. These are small and polar. So they're small enough where they can kind of weasel their way through, but they are polar, so there are barriers to it. Glucose, questionable. We'll talk about glucose in depth later. And then here are your absolute no-goes. Oh, I feel like the dotted line needs to be here, even though it really doesn't. Sucrose, way too big. Sodium ions, no way. Potassium ions, come on. Chlorine ions, absolutely not. These are large, problem number 1. Polar, problem number 2. And / or ions. That's really the biggest problem. If they have a charge, they are not getting through there. So we got our green lit fast ones. Our fast ones. We can go right on through. They're zooming. We got our yellow, slow your roll. May go through, may need to use a channel. Depends on the conditions. If they go through, they're not going through anytime soon. And then we got our absolute no-nos. Banned from the establishment. Red light means stop. You cannot enter. Turn the car around. They need to use a channel protein or active transport. Or even a vesicle. And then we have our fun little fill-in-the-blank moment. I'm just going to do this all in one color because it would be doing the absolute most if I switched every time. So generally when an atom or a molecule has a charge, which means it has lost or gained an electron, like ions, to form an ion. It's very hydrophilic and very permeable. No, I'm sorry, I lied. And very polar. And therefore not membrane permeable as it interacts too much with the hydrophilic and polar phosphate heads. It requires special channel proteins to move through the membrane. When a substance is uncharged, but still polar, the membrane permeability depends on the size of the particle. Small particles like water or ethanol are permeable. Large molecules like sugar, such as glucose or sucrose, are impermeable. Molecules such as gases, CO2 and 2O2, are very small. And because they are nonpolar and hydrophobic, they're very permeable to the membrane. Again, hydrophobic small molecules, green lid go. Small uncharged polar molecules, yellow light yield. Big hosses, no way. Ions, No way. Alrighty. We did these ones in class. The slides are in the announcements. Skip. Flip. Alrighty. So now we're gonna look at diffusion, but not regular diffusion. No. Facilitated diffusion. And protein channels. And so facilitated diffusion is basically diffusion, but through a protein channel. It's still passive transport. Where's the gray? It's still passive transport, so no energy is required. And most of the channels only allow one type of molecule to go through them. The most common ones are ions. All substances which are fat soluble, so nonpolar hydrophobic, will just go through the membrane regular, while large, somebody's calling me, polar hydrophilic particles require transmembrane proteins for transport. And that's part of what makes it selectively permeable. And so we have facilitated diffusion with our membrane protein. Oh my gosh, whoever's calling me, please give up. It's probably my brother, because it's very late, and I'm not at home. And then simple diffusion. Oh! No, this one was purple. Okay, phew, I almost panicked. I almost had to restart the whole thing. I want to at least get through the passive transport types. And so your simple diffusion, I hate that they have the similarities with a line. That's okay. So simple diffusion, things just about simple diffusion. Stop it. Facilitated diffusion, things just about facilitated diffusion. And then the similarities we'll write in the ugly gray, because that's the passive transport color I chose. Alrighty. So they both involve the movement of molecules. They're both passive transport. So no ATP. Why did I write it like that? What is that? That's supposed to be ATP. Okay. They both move substances along concentration gradient. And then movement can be in or out. It's like two ways. For simple diffusion, it is your fat soluble, nonpolar, hydrophobic, small things. They pass directly through membrane. And there's like no additional steps. Like it just kind of happens. No additional steps. For facilitated diffusion, it is hydrophilic or water soluble, / polar, large things, they pass through channel proteins, channels are protein specific, are molecule specific, can be closed, may need a ligand or charge to open. Similar to like what we saw with the action potential, how they had to be opened by the charge or the ligand. It happens, so they can be controlled, unlike simple diffusion, which just kind of happens. So we have this little graph. So, will this please? Throwing things around my desk is making a problem. So simple diffusion is this like, oh, let me not use that side. Is this like straight up arrow here. So as concentration increases, your rate of diffusion is going to increase. Although you have facilitated diffusion, where it'll increase, 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 and then level off. And this is very similar to like when we talked about enzymes and why enzyme graphs kind of level off. So simple diffusion, please. It can happen anywhere on the membrane, like it doesn't need anything else. Gradient increasing will always increase rate. Versus facilitated which is limited by number of open and available channels, or I guess channel proteins. It'll speed up until all are working at full capacity. It's like the I Love Lucy boxing chocolates phenomenon. You can think about it as like simple diffusion is if you go to a store and you don't want to buy anything, you can leave as fast as possible. Facilitated diffusion is if you go to a store and you want to buy something and it's not very crowded, very crowded, then you have to wait in line. And so like the speed at which people can leave is then limited. Whichever one works for you. I keep getting spam called by my brother, so I think I'm going to have to stop to go home before I can do osmosis. So I'll make a second one. It won't be out till Monday because I can't film them at home. Sorry.